Hey, Katarina.
Hi, Victoria. So happy to see you. Feels like forever.
Yes, agree. Hey, Jamie. Nice to see you.
Hi, Victoria. How are you today?
Well, hey, Serena.
Hello.
Hi, Serena. How are you?
Hi, good. How are you?
I'm good, thanks. I'm good. Good.
Hello, everyone.
Hello, Ethan.
Hello.
Hello.
How is everyone? Hope everything is good.
Crazy day so far. Let's round it out with some evolution of abstract thought.
I mean, it's great news, but
It's a good crazy.
Exactly. Totally good. I'm very happy to hear that. Hi, Ethan. How are you doing? I feel like whenever I've been like 2 days on Clubhouse, feels like an eternity. I didn't see people.
Oh, good. I've been getting over some back troubles that I've had the last 2 weeks. So I got a bunch of stuff done today, which was good.
Oh, yeah. I hope I'm glad you're starting to feel better. It's very annoying.
Yeah, this is good. I've been reading some other books related to this kind of topic recently.
Really? What have you read?
Right now, I'm reading a book called Incomplete Nature by a guy named Deaconess. And then,
Cool.
Yeah.
What's it about?
It's kind of a broad-based rambling about this topic and consciousness and evolution and teleology, non-teleological explanations.
Broad-based rambling, huh?
Yeah, that's how I feel about it at the moment.
That's really cool. I guess you're all ready for the talk then.
And then the next book I think we're going to do is one that I love, which is dear to my heart. It's come up many times in different ways, in different rooms. Is Gerald Edmonds' one of his more recent books, Pop Sci of his theories.
I'm not familiar with him. Could you fill me in?
Hi, welcome.
Can you hear me? Okay. Oh, wow. Okay, that might be better. Can you hear me now?
Much better.
Yes,
okay, sorry.
Welcome, Dr.
Yeah, I was on the wrong thing. I was listening instead of actually participating to the presentation. I'm sorry about that.
Welcome, Clement. That was quite an entrance. That was very dramatic.
Yeah, excellent.
The sounds of abstract thought.
How are you this
now
evening,
we got a
Doctor?
sound. Okay, that's perfect.
Well, actually, we had the guest speaker here a while ago. I don't know, Serena, if you were there. Joe Barnaby. He records brain activity from humans and as a side project turns them into art, so in soundscapes.
Oh, really?
Yeah, like when people meditate, when they are on LSD and focusing and in different states. So yeah, we should invite him back. But yeah, it's I'm not sure if it sounds similar, but would be interesting to play one day. Well, welcome, and thank you for making it, for going through the trouble to join Clubhouse and everything. Really, we are very thankful that you.
No, thank you for inviting me.
Hi, Denise. Meet our member, like team co-moderator Victoria and Jamie and Serena, Dennis and Ethan is here very often too. It will be a great discussion, I think. So really looking forward to it. And
Even just the
maybe
title of the speech is fascinating. Yeah. Oh, sorry, I'm just too excited. Sorry, I'm going to be quiet.
I agree. So, yeah, shall we start? Is everyone okay, ready to start?
Strap in, everyone.
Great. Let me start by welcome everyone to Science Society, and a special thanks, of course, to Clement. Am I saying it right? Clement Garin or?
Yeah, it's not far. It's Clément Garin. It's not far.
Clement Garin.
Yeah,
Okay.
you got it.
I'm from Europe, but I cannot speak French. I'm very sorry. My parents made me study Latin, and then at English and later on Spanish. So I'm really sorry.
It was already really good. Don't worry, I heard worse than this pronunciation. Far from that.
Yeah. So welcome everyone, and we are very excited to meet our guest speaker. And let me tell you a little bit about our guest speaker here today. Clement Garin. Sorry, I did it wrong.
Yeah, that's very good. That's very good.
He's a researcher at Vanderbilt University, and he did his master in science in Lille, in France, and his pharmacist doctor at the UFR University in Champagne, France, and his Doctor of Philosophy in Neuroscience at the University of Paris in France. And he did his, he worked at different institutes. Before he was at the Vanderbilt University. He was at Wake Forest University in North Carolina, and he published quite a lot of very interesting papers and received some honors and awards. And we are very honored to have you here today and share your exciting, really cool research with us. And if it's okay, Victoria will ask you a few questions before you start with your talk today. Thank you.
Course.
All right, here I am. So I'm so happy to welcome you here to Clubhouse and to Science Society, Clement, and to give a little bit of maybe a well-rounded introduction to our audience of you. I have a question for you, which is, if you can reflect on your life and think of when there was maybe a time that you noticed that you felt a special connection to science. And that could be any time in your childhood or young adult or any time at all, an experience or person or class, but sometime that science was a thing that you were really feeling connected to.
Well, that's a deep question. Honestly, it's difficult to tell you when I really felt the connection for science. I believe it was somehow always here, and I would say the real connection with science was probably just loving mammalians in general, loving animals, on wanting to understand them. And I guess this is the start of the big adventure, to be willing to understand how they think, how their brain is functioning, on also how they define us. So yeah, this I think this is the start of why I like science, and why I want to go into that direction. Not sure if I answered your question.
You did. You did. It's always, yeah. I'm just thinking about your answer because it's so. Yeah, I appreciate the reflection, and I appreciate your honesty and sharing that with us, because it's fascinating to hear everyone. Everyone's connection is so different, and some people feel and driven to solve a problem, and I think our answers tell us, how we connect with science talks about, ourselves too.
Yes.
And so if you can continue this question, can you lead us along the path to your current research, how you wound up here?
Sure. So I have a very, I would say, difficult, maybe not difficult, but very lost beginning. I started by willing to be, but I wanted to be a forensic scientist originally. So I have a background in toxicology, in forensic toxicology, but it appeared that in France it was difficult to have a position in forensic toxicology, and they were asking a PhD for that. And I tried to find a PhD in forensic toxicology, and I never managed to find one. And I actually find one in the lab in Paris where I was. And my PI was working on Alzheimer, and he was trying to induce Alzheimer in a mouse lemur, in the first mammals for which I characterized the resting state network. I'm going to talk quickly about that. Um, yeah, the original project was not at all in, like, the purpose of the project was just inducing the disease in this animal, and trying to understand how the brain is going to react to that. I mean, this project was like very, very difficult, and we never actually managed to reach the end of that project. However, I was also using this technique, the technique of bald imaging, which is called also fMRI, for those to know. On this technique, we managed to apply it to mouse lemur, and I, for the first time, saw the network of the animals, and I was absolutely fascinated by them. On little by little, I wanted to understand how this network are organized in other mammalians, and then I wanted to try to compare them, and then wanted to try to build evolutionary theory based on that. So yeah, this side project opened a complete door to a new world that is almost unexplored. On when I found that, I was fascinated by it. Now I'm moving toward non-invasive technology. I'm about to move back to France. I recently trained my personal dog to go wake in an MRI machine, and I managed to extract her defaultment network. Now I want to try to work with a wake animal who are willing to go in an MRI machine, and try to understand how also different breeds, how their brain is different. A lot of idea, but always related to brain function onto mammals. Yeah, it's a long story.
Thank you. That's so much. It's so interesting. I'm writing it all down. It's just so fascinating. It's like beads in a necklace, and I don't know, or stepping stones towards something really great. And so, thank you. I appreciate that so much. We all do, because I know you weren't expecting it. So
No.
now, You're welcome to launch into your discussion, and however you would like to do it, you may. We're here to handle guests and the chat in the room. I don't know if you noticed that, but some guests prefer to give their talk and then have a Q&A following. Some
Mhm.
guests prefer to have questions along the way. So whichever way you would like, we're happy to be here to facilitate that. And thank you.
Yeah, I'm completely open to any question anytime. Like, don't hesitate to stop me, especially if you have something that you don't understand. Sometimes it can be necessary to understand the following step. So I prefer that if people, is there something that I even like didn't explain well, or someone didn't understood, whatever the reason is, like, don't hesitate to tell me, oh, I need some explanation for that, and I will try to do my best to help you guys following me.
Well, thank you. So the mic is yours. And
Great.
you do have your slides, we have your
Okay.
info up at the top so people can follow along.
So I'm gonna say next slide to tell you guys that you need to move slides because you can see mine obviously. Yeah, so this article is dedicated to the study of the evolution of an important network in the human brain that is called the default mode network. And as it has been explained, this study was conducted in mostly Wake Forest University in North Carolina, but now I'm in Vanderbilt. So next slide. So on this slide, you can see that there is several representation of the mammalian brains. On the right, you can see that several characteristics has been characterized in those mammals. On one of them can be, for example, the sulcus on the gyrus, and you can see that there is a huge diversity of those shape of brain, but also you have in red the number of neurons, and you can see that there is a tremendous variability of the number of neurons. If you're interested into the study of number of neurons, there is a few amazing study that has been performed by Herculano Husel at Vanderbilt University too. She has also an amazing TED that you might be interested in seeing. So these parameters are several parameters that can be almost directly observed, but you have also other parameters such as the localization of the various areas, such as the motor area, the visual area, whatever it is. But we managed to, until now, characterize few of them, and we observe that they're not always exactly at the same location, but we still don't know why. On that some are bigger than other, and we don't know why too. It's obviously one of the things that we might thought it's related to cognition, but we need to go further to explain the mechanism that led to the actual cognition of the human brain, but also to the actual cognition of other species' brain. I still believe that it would be not only interesting to understand how our brain works, but the brain of other species. So you can move now to the next slide. So the networks, the history of the networks, the way I characterize them and the way that it has been characterized is fairly recent, and it has been really well developed thanks to a technique that is called BOLD or functional MRI. And this technique basically allow you to indirectly measure the variation of blood oxygen in the brain. So we know that those variation, they indirectly correspond to variation of neuronal activity. On that, then we're gonna record them, and we're gonna try to, for example, analyze a correlation between various area in the brain. We're gonna be able to recover an organization that correspond to, for example, the cytoarchitectonic organization, which is the organization of the cells in the brain, and that is meaningful in a numerous pathology. For example, with deafness, you're gonna observe a decrease of the correlation between the left hemisphere auditory cortex and the right hemisphere auditory cortex. So this is, for example, for the simple, I would say, relationship between bold signal and pathology. But there is one network that is actually particularly interesting because this network is related to numerous diseases that are actually a very big problem for our society right now, such as Alzheimer. We know that more and more people are getting this disease. We still are not able to really explain why does it occur. And we know that, for example, the default-mode network is particularly impacted in this disease. So we can observe decrease of the correlation between components of the default-mode network. On the function of the default-mode network, the basic way to define it is that this is the network that is going to be the most active at rest, so when you're not doing anything. So the network characterization, next slide. I need to get used to that. Next slide. So the network characterization has been performed actually in very few species in comparison to the number of species or even mammals existing on the planet. So you can see that here it has only been characterized in 16 species. All of those species have claimed to have found a network architecture that could correspond to the default mode network. But now, next slide. I don't know if you can see that. I should not have done that, but anyway. So the basic characteristic were that they all possess a motor, a visual, and a subcortical network. As I said, all the authors claim to have found a default mode network. However, several questions did remain concerning how this network is organized in human in comparison to other mammalian species. And now you should arrive to the next slide that is called cerebral resting state network in humans. Hopefully we are all on the same slide. So how is organized this network in human? Because if you want to compare it with other species, you need to know how it's organized in human. So you have basically 5 clusters that are strongly correlated together. One is the posterior parietal cluster. Another one is the temporal cluster. Another one is a posterior cingulate cortex cluster. And the last one is a medial prefrontal cluster. So 2 of this cluster, the posterior cingulate and the medial prefrontal cortex, are very important characteristic of this default mode network. And the medial prefrontal cortex is especially interesting because this network has been considered as a regulator or gateway function of self-representation. So we believe that the connection with the posterior cingulate cortex is especially important, especially since the function of the posterior cingulate cortex is known to, I would say, to allow you to interact with your external environment or your internal environment. And more you're going to be able is going to also be very important for suppressing external activity in order to focus on some internal activity. So these 2 clusters of the default mode network are extremely important for human cognition. So you can move to the next slide. So here is the first paper that I was talking about that I published during my PhD. So we had the luck to have an extremely powerful MRI, an 11.7 Tesla. And to give you roughly an idea, in human, usually we use 1.5 or 3 Tesla. So it's almost 10 times stronger, more powerful than a human MRI. You can move to the next slide. And here is the 6 network that we managed to extract in the mouse lemur. On the first thing, so if you move next, you're gonna see the DMN-like thing with the DMN-like question. We identified 2 networks that we were not sure whether they should be attributed to default mode network-like function or not. We find other networks that were quite obvious: the somatomotor network, the occipital network, the 2 subcortical network, and these 2 networks for which we have had absolutely no idea to what they could correspond. You can move to the next slide. It's called the default mode network in marmoset. In this slide, it's a fairly recent study that has been published about roughly the same time of our mouse lemur article. on what they find in this study is that in marmoset, the medial prefrontal cortex was not connected to the posterior cingulate cortex. Oh, sorry, my cat is screaming. So they study the network of marmoset with fMRI, but also by injecting tracer in, for example, the posterior cingulate cortex, and they confirmed that it's not only the correlation of the neurons that are not connected between the medial prefrontal cortex and the posterior cingulate cortex, but also the anatomical connection. So this article drive me to the article that I'm going to talk about, an evolutionary gap, because I thought, wow, this network are actually very similar to what I observed in mouse lemur. And it was not clear whether this default network organization in marmoset was just something specific to this animal or whether it's something that could be reproduced in other non-human species. So now you can move to the next slide. So now we are on the Cell Report article, and we're going to move again to the next slide. So in order to compare the network organization between non-human species and human, I had to gather several data sets of bald fMRI data. And I managed to, by collaboration and contacting people, to have researcher in Canada, but also here in the US and in France, sharing awake data sets in marmosets, in macaques. I took my anesthetized data sets of the mouse lemur, and I also managed to download fMRI data from human online. If one day you want to see how it works, there is a lot of data sets that you can easily download and play with. So we're going to move to the next slide. So this is basically what I was saying. This is a data set that we gathered for this study. And you can see that, as I said, the macaque and human, they usually are recorded on 3T, 3 Tesla MRI. And for marmoset and mouse lemur, which are very smaller species with smaller brain, you need very more powerful MRI to study them. We can move to the next slide. Here is the network that I extracted in those four species. And I used to do that an algorithm, a machine learning algorithm that is called dictionary learning. So this is a very fancy method, but I actually like it because in comparison to other methods that are traditionally used in the field, it actually allows to extract networks that can be attributed to it allows you to extract region that can be attributed to different network. So this is actually very important to me because from an anatomical point of view, it makes sense that a region is not only connected to one network. But anyway, I applied this algorithm and I extract the traditional default mode network organization that you can see on the top left of the human. So you can see the PCC component, the medial prefrontal component, and the temporal component. I extracted using the dataset from Canada of marmosets on the right, the default mode network that correspond to exactly what has been described in the paper in marmoset that I showed you earlier. I extracted the macaque default mode network in an awake dataset that has been shared by a French team. And you can see that in comparison to human, for marmoset and macaques, we don't have the medial prefrontal component, but we still have the PCC component, the posterior parietal component. And we have actually a very strong component that is located in the dorsolateral prefrontal cortex that we can also find in marmosets and in mouse lemur. And you can see that the mouse lemur also have this organization without the medial prefrontal cortex. So this was fairly surprising, but also not that much given the literature that we had. The interesting thing was these common clusters, dorsolateral prefrontal cortex. This cluster is actually including regions that are dedicated to vision. So now we're going to go through the hypothesis. And again, I'm going far from the data, so it's just an idea. My idea was that rather than having a species that is gonna, when they're gonna have, I would say, free time, or when they're not going to have anything special to do, they're going to be focusing on the external environment. They're going to see if a predator is going to come and try to eat them or whatever. But they need this connection with the PCC in order to focus on the task, which is looking at the external environment. To me, my idea was, if the medial prefrontal cortex is strongly connected to the PCC region, it's because when we have free time, we're trying to planning the future or to think about how we're going to do this or do that. But this is just hypothesis, so please, nothing is like proved yet, except the organization that you can see here. So now you can move to the next slide. So the thing is that if the medial prefrontal cortex is not involved in a similar network as human, it must be involved somewhere, because we know that marmoset, macaques, and mouse lemur, they have a medial prefrontal cortex. So to what region is this network connected? And we find that it seems to be consistently connected to a temporal region that is not the same across all those three species, but the characteristic is that it's connected to a temporal region. We can also see in macaques several connections with the PCC, but that are less stronger than with human. So maybe there is a beginning of development of this connection between medial prefrontal cortex and PCC in macaques. We can move to the next slide. So please press 3 and you're going to have the full slide. It's going to be much easier. So here there's basically another method that we use in order to extract the connectivity, basically just the correlation between the different cluster of those 4 animals. And so I'm just to be sure, I am on the slide that's called connectivity difference between hominoid and humans. And what you can see here is that if you take the pattern of connectivity of the PCCs on the top left, you can see that the connectivity profile of macaques, marmoset, and mouse lemur are way more similar between themselves than when compared to human. And if you look at the connectivity pattern of the medial prefrontal cortex, you can see that this connectivity pattern is also much more similar between non-hominoid species, so macaque, marmoset, mouse, lemur, rather than when they compare to human. And after, we've done fancy statistical analysis, and we also confirmed that it's just not only something that you can see visually, but also something that you can prove statistically. So we can move to the next slide. And basically, this paper, from that point, is trying to reproduce with different methods and with different statistical analysis the result that I explained to you since the beginning. So here, for example, one of the things that is extremely difficult to do in interspecies comparison is to directly compare the ball signal across species. And the reason being, I was talking about all these different field strengths between MRIs, but also all these different brain sizes. The reason being is that it's going to generate very important signal-to-noise ratio difference across all the species. So one of the ways that I choose to directly compare the signal was to internally, within a species, compare if the signal between the PCC, the correlation between the PCC and the dorsolateral prefrontal cortex, and the correlation between the PCC and the medial prefrontal cortex, were higher than another within one species. So you can move completely to the next slide. So press 2. And now we are on pairwise comparison of the relative correlation differences. This is what I was explaining. I can't compare directly the correlation across species, but I can see if one connection is higher than another in species, within a species. And you can see that, for example, if I take the dorsolateral prefrontal cluster correlation to the posterior cingulate cortex, so this is the extreme left on the figure A, this connection actually less stronger than the PCC correlation to the medial prefrontal cortex, so the blue box. And if I reproduce this analysis in other non-primate species, I can see that we have the opposite relationship. So this is why I was talking of a gap. It's because I believe that something happened in the evolution that allow the medial prefrontal cortex to be strongly connected to the posterior cingulate cortex. So you can move to the next slide. I was wondering whether this correlation was affecting only the connection between the medial prefrontal cortex on the PCC, the posterior cingulate cortex. And to do that, I took another region of the default network, the posterior parietal cortex, I analyzed the correlation between posterior cingulate cortex, posterior parietal cortex, on dorsolateral prefrontal cortex, on medial prefrontal cortex. So this is basically the same analysis as earlier. Instead that I replaced the PCC by the posterior parietal cortex. So now you can move completely to the next slide, and you can see the result of the PC with the dorsolateral prefrontal cortex that are less higher than the PPC with the medial prefrontal cortex in human. And we have the opposite relationship in all the non-hominoid primates. So one of the things that what I wanted to clarify was most of the study that study the networks in various mammal species use anesthesia. And I was wondering, okay, why nobody saw that before? I mean, especially in macaques. And I had an awake on an anesthetized dataset in marmosets. Then I just wanted to see if the anesthesia would strongly affect the correlation between this region in order to see if it could explain why it was not so obvious before in earlier study. Now you can move to the next slide. And you can see that, yes, the effect is absolutely huge, that the correlation, whether it is between dorsolateral prefrontal cortex, so 8A, on PCC, so 23B, that we have a strong decrease during anesthesia, and it's exactly the same thing between medial prefrontal cortex and PCC, despite that this decrease of correlation is actually much less than the correlation between dorsolateral prefrontal cortex and PCC. So that would explain why we or other researchers struggle to differentiate the connectivity between dorsolateral prefrontal cortex and medial prefrontal cortex during anesthetized session of bold recording. So now you can move to the next slide. So in conclusion of this study, we observed that the medial prefrontal cortex is poorly involved in the default network of non-hominoid species. That you can press the correlation between medial prefrontal cortex on the whole posterior default network is weaker in non-hominoid. On that, we potentially discover a new network that seems to be specific to non-hominoid species. We still need to define what could be the function of these networks. But after discussing on also reading a lot of literature, again, this is an hypothesis. This network could correspond to some social function of the species. But again, we need much, much more study before arriving to confirming this hypothesis. So this is the end of this talk, but if you are still willing to hear me, if you press next, a very recent article that is about to be published in PNAS. So if you have time, I'm willing to explain you that. It's going to take roughly between 5 and 10 minutes. So please let me know if it's okay for you.
Yeah, it's totally okay with me. It's more if it's okay for you to talk longer. Yeah.
Yeah, of course. I mean, I'm happy to give you that pre-publication and to it's actually very related to what I'm talking about. It's almost like a continuity. So okay, but it's gonna be very quick. I made it fast. So So the article is called "Multi-Level Atlas Comparison Reveal Divergent Evolution of the Primate Brain." So one of the questions that is fairly common in the literature is whether this medial prefrontal cortex is overexpanded in human in comparison to other species. And there is a huge debate with a lot of good research involved that was claiming, yes, it's overexpanded in human. No, it's not overexpanded in human. So I tried to dig a little bit into that question. And to do that, you can click on next slide. I actually downloaded freely available online digital atlases of the mammalian brain. And I find 18 different atlases. And in these 18 atlases, I discovered that everybody is using a different classification of the brain area. So I had to work into reclassifying this area into common classification. So you can click on next slide. And this is the reclassification of all these 18 atlas in different level. And those level, for example, the level 2 is the lobar level. So you're going to have in red the frontal lobe, in blue the parietal lobe, in green the temporal lobe, and in purple the occipital lobe. The level 3 is more dedicated to regions that can be associated to function, such as motor, sensory motor, or medial prefrontal cortex, etc. So what I've done is that I wanted to study whether the frontal or the medial prefrontal cortex were overexpanded in human. And you can click on next slide. So the traditional way of analyzing that is to perform what we call PGRLS analysis, which is an analysis that is going to take into account the genetic proximity between species and is going to try to draw a regression between here the volume of the different lobes of the different animals. And what we can see here is that on the top left corner, the first the first plot, you can see all the that all the primate species are indicated with an arrow, and that the macaque, chimpanzee, and human, they are indicated in red. And what you can see is that the regression line between the parietal lobe volume and the occipital lobe volume is actually the slope is actually much higher in those species, macaque, chimpanzee, human, than in all the other mammals. On mouse lemur, which is a primate included, but also marmosets. So the thing is that we started to have an idea of, those volumes seems to strongly increase in primate, and it just didn't appear like suddenly in human. It's a slow process of evolution. On the one of the lobe that seems to be the most overexpanded in human is actually the parietal lobe. We reproduce this analysis with the frontal lobe, and we find that the frontal lobe was also could be considered as overexpanded, but was also following the same pattern as for the parietal lobe. On the two other lobes that were with lower slope, in again those species, so macaque, chimpanzee, and human, was a temporal on the occipital lobe. So those lobes contain actually fairly amount of we'd say sensory or just visual auditory area that seems to be less expanded than the parietal and the frontal lobe. So we created a classification of the slope, and we find that the parietal and frontal lobe were the lobes with the highest slope, and then we have the temporal and the occipital lobe. And again, I'm talking about the catarrhini primates. And if I reproduce the same analysis in the other mammals, I found that the occipital, the temporal lobe are the lobe with the highest slope, and then you have the frontal and the parietal lobe. And we find that the frontal and the parietal lobe, they always follow each other, meaning that it seems that there is an interdependency of those two lobes. When one is growing, the other needs to grow, whatever the species it is. You can click on next slide. So this analysis that I showed you earlier is a traditional way to do it, but there is actually some problem with this analysis that we think that we solve partially. For example, you need to use one region as regressor. This is a problem because if you use a region that is going to be underexpanded, for example, this is going to give the impression that another region is overexpanded. So we created this matrix that is able to analyze all those overexpansion between every pair of region possible, and then you have the result in a matrix form. And we confirmed that in human, the frontal, the parietal lobe, the medial prefrontal cortex are overexpanded, whatever the kind of atlas you can use. So here on the left, you have the human atlas of Glasser, on the human atlas of the BrainTome atlas. So, and we find that, well, I'm not going to show you that, but in chimpanzee and in macaques, we also have the same pattern with a frontal lobe that is considered as overexpanded. So showing that this is a very long story of expansion. And if you use other species, for example, marmoset, you're not going to observe that the frontal lobe is overexpanded in these species. So you can click on next slide, and it should be the conclusion. Hopefully I didn't forget. Maybe I forgot to tell you to change that. Anyway, so now we're on the conclusion. My conclusion is that I consider that humans are special because they carry the most relatively enlarged frontal and parietal lobe in an infraorder, the Catarini, that exhibit a disproportionate expansion of this area. So we think that we had a divergence in primate evolution, roughly in our common ancestor of between old world monkey, great apes, and non-hominoids. And as I said, the parietal lobe is systematically linked to the frontal lobe. If you want to know the consequences on the functional architecture, you can read the cell report, but we already talked about it. And now I'm trying to, as I said, in the tradition, to more and more go in the mechanism that could explain this overexpansion, this new connection in human, and how they can lead to our cognitive abilities, but also to other cognitive abilities in other species. So finally, you can go to the next slide, and I would like to acknowledge all the people that participate in the study in Vanderbilt, Wake Forest University, at the CEA in France, and at the Institute of l'Institut des Sciences Cognitives, Marc Jeanrod. I have to tell it in French. I have no idea how to translate that. But anyway, thank you very much. And now the floor is open to questions.
Thank you so much. This is, this was such an amazing presentation, and special thanks to that you guided us through the whole story through your papers, and this was so much work. I can't imagine. How many years does this presentation summarize in work?
So if you take from the mouse lemur, this study started in 2017. On the cell report on the PNS, I started in 2019.
Yeah, well, and and thank you that you're sharing such a brand new paper.
Yeah, it should be out next month.
That's
I should
amazing.
receive the proof like tomorrow or the next day, so it's going to be out very soon.
Well, congratulations. That's amazing. And that's really wonderful data. It's really cool to see basically the pathway to like the incremental steps to the human brain. That's really interesting. But I want to give people opportunity to ask questions. Serena, please go ahead.
Well, wonderful talk. Such a broad range of data, and I love how the perspective of tracing the differences of evolution of the brain functional regions, but also the insights of how you're looking at what, well, in the default mode in this case, but what regions correspond to what over the evolution in the species. I'm curious. You closed on looking into the mechanism. I've taken great interest in astrocytes recently, and the function of the astrocyte syncytium and how it relates to coordinating neural activity. I'm curious. In some of the readings I've come across, the proportion of astrocytes to neurons and how that's changed in evolution. Um, for example, there's 1 to 3 astrocytes in mice and rats, but by the time we're to humans, there's 3 to 2. So there was a period, and I don't have corresponding data from from chimpanzees, but there is a suggestion that our astrocyte population grew in complexity and numbers. I'm curious, have you looked looked into astrocyte functions in the role of connecting these different regions, and what comments you might have on that?
Yeah, actually, you just taught me something. From what I read in the literature, I thought that the supporting cells, other than neurons, were fairly stable in the brain. It's Herculano Husel who produced a paper on that. But I mean, I think she just studied neurons versus other cells. So it's possible that there is subpopulation of other cells that might have changed during evolution. So I might on the thing is that with ball techniques, we are mostly studying what would correspond to neural activity, and we have absolutely no idea of what could actually participate to that. And you're right. That's why I believe we should go deep in the mechanism that sustain that. And I have few idea of how to do that, because the big problem with this kind of characterization is that it needs to involve a lot of species. On From what I understand, it needs to involve the whole brain. So basically, if you want to try to characterize that with classical histology, it's impossible. On the technique of Herculano Husel is very interesting. She basically take brain sample, extract the cells, count them, and then tell you whatever population you have. But you still don't have a very good spatial resolution of how the brain is organized. However, there is some other MRI technique that could be applied that could help us answering that question. But I think you are completely right. This is a very, very important question on not only neurons should be taken into consideration in order to understand how the brain has evolved.
Also curious on the fMRI technique as well. There's you noted that it's an indirect indication of neuronal activity,
Mhm.
and there's some question. I'm certainly curious about interpreting the signals in the presence of these astrocytes. They do connect through the blood-brain barrier and to each other through gap junctions and ultimately to the synapses. And they communicate through these calcium resonant waves that may be more energetic in demand than actual neural firing. And I'm wondering if part of what we're seeing in the MRI signal is the activity of supporting those calcium waves in addition to the neuronal activity. I'd be curious if the signal is conflating the activity of the two.
Oh,
In,
yeah, I think you're right. I think it's possible that some other cells are actually involved in the network consumption I mean, that we are, yeah, the network consumption that we are registering with uh, with the fMRI technique. But there is also supporting evidence that tend with electrophysiology or
mm-hmm,
with PET that also tend to show that those neuronal activity are generated by neurons. But again, it doesn't tell that part of the signal could be generated by astrocyte. That
mm-hmm.
it's difficult to prove, but it would be very interesting to try to have a functional signal that would be specific to astrocyte. But there is some techniques that start to appear in the field that in the long run that could lead to answer to this question.
So there's a final question. It blew my mind. If it was a 2012 paper done out of Rochester, Nedergaard's group, I believe. They took human astrocytes and progenitor cells and grew them in mice. And the results were fascinating in the sense that the astrocytes did take well. These were uh, immunoinsensitive mice. They took well, they took over and integrated with the synapses of the mouse neurons, and were much larger. They propagated waves faster, and the mice did in fact have far superior performance in learning, and they were smarter.
Mm.
Um, and so the distinction between human astrocytes and the astrocytes of mice has grown in complexity with the size, the number of processes, and the kinetics of the waves. I'm just curious how much of a component in light of your topic, um, not just increases in numbers of astrocytes, but in terms of complexity and performance, might also be a factor. I thought that was fascinating.
Yeah, I think this is great remarks, and I would love so much to be able to answer to this question, but it's this is what I wanna study in the close future. On hopefully, if we can have another talk in 15 years, I could provide some answers. But yeah, I'm not yet there. I don't think the literature in terms of evolution is, from what I know, there yet. But yeah, I would be actually interested if you can send me this reference. I would be actually very interested to read this article.
Oh, certainly. I'll follow up. Certainly.
It's a
Thank you.
Thank you
pity
very
that
much.
you're not in Canada, but anyway.
well, yeah. No, we're
And
gathering together some resources to look at modeling of these astrocytes and perhaps have an impact in AI. So yeah, I'll certainly follow up. Thank you.
Thank you.
And can I thank you for an amazing talk, Doctor? This is incredibly interesting. And there's actually a question from one of our audience members who asks, out of all the books on your bookshelf, which book do you treasure the most on this topic?
Oh, I'm really bad with name, but I actually got the book near me, so we can help you with that. It's called Evolutionary Neuroscience, and it's edited by John Cass.
Perfect. Thank you very much.
Yeah, if anyone has any questions, please press your mics. Ethan, do you have a question, a dolphin-related question, maybe? Maybe Ethan is away from his stuff because
No,
he.
no,
Oh, there you are.
nothing in particular.
Okay. Yeah, because Ethan, if you realize, come on, he has the dolphin brain there, and I know he read a lot about dolphin brain. So, are you planning on one day studying also the dolphin brain? Probably quite
I
simple.
honestly thought about that. I thought, like, yeah, I thought it would be amazing to put a dolphin either anesthetized or awake in an MRI machine. I thought of many way we could do that, and I think it would be passionating. Like, do dolphin have something that looks similar to a deformant network? On like just their brain is, I have no idea how their brain is organized, and I believe that we know little about that. I don't know, maybe Ethan is going to tell me I'm completely wrong. It's totally possible. I do not know anything about
uh,
that, but
well, one thing that's interesting about it is they have a whole extra lobe. But in general, like a lot of the, when people do these comparative analyses and they create sort of a hierarchy, and
mhm.
human brains are usually always on the top. So I know there's been like brain size with cephalization taken into account, and neuron density, types of neurons.
Mhm.
But in actually almost all of these categories of comparative analysis, dolphin brains are actually above human brains in almost every category, which is just kind of interesting thing when you see this kind of work done to sort of characterize various features of complex, quote unquote, complexity of brains.
And is there an atlas of the dolphin brain? Is there, do we know if they have like motor area, like roughly in the same
Oh
position
yeah,
as
yeah,
us,
yeah,
or
there's a lot of it. There's also probably a lot that is, well, the Navy does a lot of the research, so a lot of it is I think hidden inside there. But there's definitely atlases and that sort of thing. I'm not a super expert or anything. It is just interesting to see that when categorizing a lot of these different quantities in terms of trying to get some kind of semblance of hierarchy and these various things. It's an interesting pattern.
Yeah, thank
Yeah.
you for that.
I mean, you can
Doc-
see just in, like by looking at it, there's more. One of them is counting the density of gyri and sulci, right? And human beings come up high. But if you look at it, you can see the density is even greater in dolphins for that major morphological feature alone. But then the types of neurons, the branching of dendrites, all these kinds of things that people have done, are comparing the neocortex to body size, the ratio to that. Again, dolphin brains, bottlenose dolphins, not regular dolphins, definitely. Like spinner dolphins don't have this property. It is typical only to bottlenose dolphins.
Yes.
Yeah, thank you so much. I mean, that was wonderful. I mean, I'm talking about dolphins as well as whatever we had here. Thank you for sharing your fascinating work. I mean, I think that from the clinical perspective, I'm thinking about some of the patients, especially that they are dealing with the Alzheimer and dementia. Actually, as you might know, that in the MRI scan from them, they have I mean, tau tanglement and as well as all of those symptoms that is showing the changing in the brain region and functionality, but somehow they are not symptomatic. So I was just wondering because you mentioned about the chronic pain and however we know about the microglia and which they are just inducing the LTD in that region, and there's a relationship between them and the inflammation with the TNF alpha, interleukin number 2, and also PP2P. So I was just wondering, could you gather any information in a clinical level?
Um, you mean about those factor measured in those species, like how they evolve,
Yes, I mean, I
or
was
I'm
just
not sure.
wondering because we are talking about the species, but in the same time, in a human, we have some similarities. That's why we are just using the animal model. And my question specifically was about the region that you just mentioned and microglia and plasticity as well as the Alzheimer because they can be related somehow to each other. I was just wondering maybe you have further information that you can share with us in a clinical level.
I mean, I'm sorry. I'm not sure if I fully understand your question, so I'm gonna try to answer. But if I answer, like, if my answer is not correct, please help me. Um, yeah, the thing is that what is interesting is that in macaques, in marmosets, in mouse lemur, they don't develop disease that could correspond to what we call Alzheimer's disease. They don't have naturally like tau proteins. They develop very few amyloid plaques. So the question, one of the questions, we know partially why the DNA sequence of I, I'm far from my field of knowledge now, so I'm afraid to say some stuff that are not true. But yeah, the DNA sequence is different. That the one that is coding from the precursor of amyloid, and I forgot the name. But anyway, there is some genetic differences that explain why this amyloid protein do not aggregate in those species. But except that we know that we can, despite this modification of DNA configuration, we can still manage to induce it. Um, and it seemed to spread along the connectivity, along the network, the way that the regions are connected together. So we know that this difference of connection might have an important influence on the way that the animals model that we are using are going to develop symptoms on potentially when we're going to study their behavior or their cognitive ability that might be different. And I think this is something that with this study now need to be taken more in consideration. Yes, in those animals, we can induce pathology that from, I would say, biological point of view, when you're going to measure amyloid plaques or tau or inflammation, might look like something that could be similar to human Alzheimer, but in terms of function, since the network are completely different, you might expect a different spread of those protein in the brain. So yeah, this is what I can say in terms of clinical. After, like, the way that I build this data set, it's so rare on the amount of data that you need to perform the study is so important that it's very, very difficult to answer exactly I don't know, NFT or whatever factor you want to compare across species to actually do it across species. It's very difficult because we are missing data and we are missing way of comparing those data between species. I don't know if I'm clear with what I'm saying, and if it answers your question.
Yes, perfectly. That's why I mean, there's supposed not to be clear answer because the science is not clear sometimes. But and one, I mean, another question that I have: did you had any experience with exposure with the bacterial or gram-negative bacteria in any of these regions, in other experiment or such a thing?
With what kind of bacteria? Sorry.
Gram-negative bacteria.
Oh no.
Okay, so thank you so much.
No, you're welcome.
My PhD supervisor was Joseph Ledoux. I don't know if you know him, but he did a lot of anxiety, fear related memory. And he basically later on, after all the years of us doing research and saying it's fear memory, he didn't, allow anyone to say fear memory anymore. He said only threat memory when we did studies in rats and so on. And he also later published articles and books that annoyed a lot of people. But he might be right when I look at your data. And that's why I thought your data is so fascinating. He basically said most of the stuff we did was useless because whatever we for anxiety disorders and to help people, because the treatments we came up with didn't really solve anxiety for people. It kind of downregulated general brain activity, but there's no drug or cure for anxiety disorders, PTSD, and so on. And what he says is that the brain structures for higher thought, abstract thoughts, like what people have, predicting that something will be completely wrong and everything, I don't know, you have an anxiety from traveling, let's say, and then you imagine all these stories in your brain, in your head, and then you avoid traveling or whatever it is. And he says there must be a very distinct brain structure in humans that we are not studying in animals, and that's why the drugs we came up with, and they work really well in rats, they won't work in humans, and we can all just go home. Basically, he didn't say that, but kind of a little bit. So yeah, would you support that? Because he says, it must be in the human, very distinct human networks that these worry thoughts or anxiety abstract thoughts happen in humans that cannot really happen in other animals, especially not in rats. Yeah.
Yeah, I think you're so. It's exactly the same problem for me with Alzheimer or other problems that we try to solve with mouse and rats. I think this is great to study the basic mechanism on. I think I can't count the amount of time we solved the induced Alzheimer disease in mice and rats, but never actually managed to achieve to do it in human. And it's exactly the same problem in your field. Um, I wonder if, like, the way that science is done is always the same thing. Like, we're gonna use mice or rats to whatever the pathology is. we're gonna use them because they're small, because it's easy to house, because it's easy to handle. On we don't consider the relationship between how the animal is functioning on how we are functioning. On Yeah, I believe that human are special, so it's actually, you're right, difficult to study the yeah, emotion or anxiety in human based on rats brain. But I also believe that each species is special. I believe if you try to 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 cure the anxiety in dogs using mouse on rats, you're gonna have the same problem. If you want to cure that, you actually needed to either do it in a probably genetically closer species to the one you are studying, or to do it in the real species, which is obviously not an option in human. But also, I think maybe we should also think better how we choose our model. Or I might say, this is a hypothesis on has no real base. But for example, for the problem of anxiety, I wonder if, for example, dogs would be a more adapted model to study human anxiety. These animals tend to be very anxious. They also are animals that are very social, that potentially display characteristics that could be closer to human, that would potentially help us to have more translational data. But again, all of that is linked to the fact that we don't know anything, or know very little about mammalian behavior, mammalian cerebral organization, on genetics, on how all of them are related together. How, yeah.
Yeah, and then we also keep mixing up mouse data and rat data many times, and mice are mainly prey and rats are
Yeah.
mostly predators. And then sometimes some labs mix them up in the same lab, and and the mice are constantly scared. Yeah, I think we have an issue too, like that we basically humanize all these organisms,
Mhm.
right? We don't consider how they would actually live out there in their real habitat, and that will already kind of mess up the data probably. So, but I think we're getting a little bit better at it. So, but yeah, it's interesting. So, are you planning? So, I know you're mostly evolutionary biology, like neuroscientist.
Mhm.
So, are you maybe, or are your colleagues maybe going to do more in the terms of different disorders and use that data, or
No, I'm in a lab that is mostly studying working memory with electrophysiological techniques. So I was quite alone in my field, in my lab. Um, I Yeah, I would like to. I'm actually very interested into studying anxiety in dogs, on try to actually compare how it works in human and dogs, and to potentially see if there could be um, an interesting species to help us understand how this mechanism of anxiety works in us, work in them, and try to see if there is common features that could define them for a potential therapeutic target, or whatever. But I'm not yet to that point. Now I'm gonna try to reestablish myself in a new lab, and try to begin my, yeah, my evolutionary study. And this is sadly project for that will be not developed yet.
wow, that's congratulations. Where are you going to be again?
So I'm gonna be in Lyon, in France, on in a lab that is called Institute of Cognitive Science, Marc John Rod.
That's wonderful. It's so nice in Lyon. I
It's a nice town, yeah,
yeah,
that's true.
it's wonderful, and the public transportation is wonderful.
Yeah, it is.
We have the metro every few minutes, and it's actually nice and new. Anyways,
Yeah, I will be glad to not use my car every day, that's for sure.
right? Did you miss friends a lot?
Yeah,
You did.
I didn't went back since COVID, so it has been almost 3 years without seeing my family. So yeah, I'm extremely excited actually to go back home in 1 month or a little bit more than 1 month.
Oh wow, that's wonderful. Yeah, my family is coming this summer. I hope I'll go. I'm from Portugal. Mm,
Oh, nice.
yeah, I hopefully go in the winter. But yeah, I don't know. How many minutes do we have left, or is it getting kind of late for you?
For me, I don't know. Are you talking to me?
Yeah, yeah, yeah.
Yeah.
Wanted to check with you.
I mean, I'm. It's up to you guys. If there's still a question, I'm here to answer. I have no problem. I have nothing
Serena,
scheduled.
I
So
know Serena has my question.
Well, yeah. So, okay, I'll take those extra minutes. Um, I'm curious though. What's fascinating about your data is you're exploring the default mode, correct?
yes.
And it's an interesting question to think. Well, if, in terms of thought um, across the species, what the default mode might represent, and is that line of thinking, in correspondence? For example, I doubt mice are sitting around worrying about astrocytes, for example.
Mhm.
But so it's a curious, and Katerina sort of touched on this with, are mice afraid all the time versus rats? I mean, are there any? I mean, how would you even approach controlling, categories of thought and categorizing those with your methods under the corresponding fMRI data with the techniques you applied?
You mean in human or in other
Well, across
species?
the
In human, I could
species,
have.
putting it could be, a little macabre to put, have a cross species condition that would, require certain regions of the brain for processing, and see if those regions are at least better in better correspondence. Um, do you know? Does that make any sense?
Yeah. Like one way I can think, for example, is so there is first thing is that there is very little spaces in which you can actually like study thoughts of emotion, because the first step, especially with fMRI, is that they need to be willing to go in the magnet, and they also need to be not completely panicked about the idea of being in the magnet. So this is restraining the potential spaces to, I would say, macaques, chimpanzee, um, whatever hominoid, humans, maybe dogs, dogs for sure, and maybe other species that I don't know very well, but it's very narrow amount of species. But if you want to do that into species, I believe that, for example, one way to study emotion, and it has been done in dogs, I don't think it has been done with fMRI, is, for example, to show them picture of their owner, of someone they know, someone they don't know, and then you can start to, so you will not have a direct access whether they're actually understanding what they're seeing, but it's kind of the result were kind of showing that yes, the dogs had region that were related to positive emotion when they were seeing their owner. They had other area when they were seeing someone else, that kind of stuff. I believe that we can also use the system of eye tracking to try to see what happens when they look, I don't know, more into the eyes of someone rather than the rest of the face. Um, but of course, there is no direct way to study like their fault. You can't tell to a dog like think of a bolt and see what's going on in his brain. And I wish it would be possible, but yeah, the problem is that you have very little insight in the fact that they actually did what you think that they were thinking or not.
Right, right. So perhaps through some type of difference where you've already characterized, their default modes, but in showing them a pleasureful conditioned stimulus
Mm.
versus one that isn't, and looking at the differences from their respective default modes, is that to see if there's at least a difference in activity
So
corresponds.
yeah, the thing is that you should not see a default network if they are engaged in a task. The default network is going to disappear, and the network that is associated to the function that you are using is going to show up. So studying the default network is basically studying a species at rest, which is also one thing that is very difficult. On, for example, in this study, the cell report, one thing that I want to dig into is how I can be sure, absolutely sure, that those species were completely at rest. Because this is one of the criticisms that can be made. We still can't ask to the monkey, the dog, the marmoset whether he's completely at rest. So you can, with the statistics on um, with a huge population, believe that okay, these animals are habituated since a very long time to go in an MRI machine and they're not stressed, they are calm, they don't show sign of fear, but still it's still a question that is open to me. And it's still also a question that could explain this difference of architecture. So all of that to say that, yeah, the door is not closed yet, and we still need to try to go further.
Well, what about the
I,
so putting
I
in I'm sorry,
no, go
Katerina?
ahead. Go ahead.
What? Okay, I was just going to ask if you were to look at the decay of a conditioned stimulus and the arrival of that default mode
Mhm.
through when as it wears off and they get bored and they go back to the default mode, um, there could be in principle a time sequence that would show the returning uh,
Yeah.
of the default mode. Maybe there's something there.
Yeah, that's interesting. And also so the deformer network has shown also to be, for example, not only active during rest, but there is also, so we think that mindfulness is activating the deformer network. It's what has been shown in human. So, but it's absolutely impossible to ask a monkey to perform mindfulness task. But there is also another way to studying the deformer network, which is called task switching. This could be an interesting way of tuning the deformer network in macaques, but this is still open question.
So I have two things and two people you might would like to collaborate with. So we had a guest speaker here, I think 2 or 3 months ago, Dr. Field, and he works actually for a company, but he developed a kernel flow device, which is using near-infrared spectroscopy system
Yeah,
to,
yeah.
yeah, as a helmet that you can carry around, and it works really well. So I believe you can train a lot of different animals to wear a helmet, like at least a dog and
Yeah,
like a monkey
I'm
and
actually working on it.
oh, you are, oh, right. Yeah, maybe you can. I sent you in the back channel a message, so if you go on this paper airplane symbol you see the messages, but I can also send you an email from, yeah, he was here,
Yeah, please.
yeah. And then the other guest speaker we had last Friday, he is in the UK,
Hmm.
and he works with dogs. So he's working like he just published how dogs recognize dog and human emotions, and he's a really cool dog behavior scientist. So it's
Ah,
just
yeah.
two people I thought that would be interesting maybe for you
Yeah, yeah,
to
yeah,
check.
exactly. Yeah, no, please, yeah, share me the information. I would be glad to discuss with them because the FNIR problem we actually the FNIR we have actually a lot of problem with it. So it could be nice to have another person that could advise me on that. That would be great. Yeah, for the dog behavior person, please, the researcher, please. But I
So,
can't
so you're
see
working on a
the.
helmet like this, like also using near infrared spectroscopy.
Yeah, but the problem is that the dog brain they have a fixed skull on like there is very little area that you can record and yeah
And
if yeah
it's,
on it's the infrared need to be very adapted to the dog brain. I have a student that is working on it, but it seems to be more complicated than I originally thought. So, uh, but yeah, having other
maybe there are
people,
dogs with less thick skull.
but they have like they have like excuse me, it's not really the skull, it's the muscle that are surrounding the skull that are big.
Oh, so there are no dogs with like just very little muscle,
Sorry.
like dogs that have very little muscle. You know those little dogs that like
I mean,
move.
in comparison to the size of the brain, they have big muscles.
Ah, yeah, I see, I see. Yeah, well, I thought I had a good idea. My
It is,
friend.
it is, it is. I was working on it like last month, so it's and I thought it would be great to be able to measure that, but yeah, I mean, probably, I mean, some people done that. There is one article that managed to do it, but you need to you are gonna be basically able to record only frontal area, which is nice. I'm interested in this area.
And cats, don't they have like? I mean, especially their, their
Yeah,
visual system is very similar.
yeah,
Maybe
yeah.
they.
It could be a good idea in cat. Yeah.
Moniz, I'm sorry that we made you wait for so long. Ask ahead.
Oh, it's okay. Thank you very much. Hello, everybody. I just have a question, but I'm worried because, I have two questions. One of them is related, and the other one might not be related. But I would like to know if maybe you can help with an answer, doctor. The question I'm asking about basically ADHD, and I was wondering if fMRI can be can confirm actually someone can confirm like a case of neurodiversity like ADHD especially like maybe in dopamine receptors or frontal lobe. And my other question is that I read an article like suggesting that dogs there was like a models of dogs that it was like to prove if dogs might have similar traits of those with the ADHD, and that like what I know is like ADHD, the traits of ADHD are like heterogeneous, especially even in for human between one patient to another. It's very different. And but have you, doctor, come across any model that proves if dogs have similar traits to those with ADHD? I hope my question was clear. I'm sorry.
Yeah, it is. It is. There is two questions in your question. The first one was whether we could characterize ADHD with fMRI. I have to say that I don't know, especially for ADHD, because there's been a long time I didn't look into this paper. But I know that um, for most brain particularity, fMRI can be a great tool to characterize a group, but it's more difficult to characterize individual because fMRI has an important variability on it's not yet applied in clinic because of this variability. I know that there is several machine learning algorithms that are more and more able to with by using this bold sequence, this fMRI acquisition to predict whether you have ADHD or not, other pathology, but we're not there yet. Probably like we are currently building like more and more powerful MRI. I'm not sure if it will one day be used in clinic or if it will be useful to increase the repeatability of the fMRI acquisition, or if it's come from another problem like the antenna or whatever. But yeah, we're not that year yet, but maybe in few years, decades, it will be a thing. And I really hope it will, because I think there will be a lot of clinical application on the field I know is mostly Alzheimer, and I know that having an early diagnosis for this pathology can be very useful for ADHD. I'm really not a specialist, so I can give you more information about that. On for the fact that dog could be a good model for for human ADHD, I honestly don't know. But it's a very interesting question, and you make me want to dig a little bit into that. But I can't answer right now, sadly.
Thank you, doctor. Thank you very much.
Thank you.
Yeah, we had in the newsroom this morning. We saw, the a paper, like an article about the paper, that how much ADHD increased, and that actually the medication really doesn't help much with the kids studying better. And but yeah, that's a whole nother topic and discussion. But I think it all goes back to in what animal models we studied all of these effects, and then we just translate it to humans, and yeah, that doesn't probably work. But that goes back to my previous comment. So yeah, I think, does anyone have another urgent question? It's getting kind of late. So if there's no, let me check the chat. Oh, Mona asked a question. She is curious in the chat. She's curious if the amount of social interaction plays a role in development of abstract thought in various animals.
Wow, that's a deep question. I wish I could answer. Uh, I mean, I have no scientific base to answer to that one, but I guess it does. Uh, Social interaction, I mean, especially for primate, are really important. We know that social interaction preserve our cognition. We know that social interaction help us from not getting for slowing the symptoms of Alzheimer. So yeah, it probably have a development on abstract thought, but yeah, I have no science behind that to clearly answer on to accurately answer to that question. I'm sorry.
Doctor, if a part of the brain is misfunctioning, would that be that can show in fMRI, even if in animals or human?
Yeah, it can, but it's still difficult to find it within one individual. You can see if a group of people with Alzheimer, for example, is going to be different from a group of people without Alzheimer. But within one specific patient, you can't. It's it's not
So it's
used in
like a
clinic yet.
comparative way.
Sorry.
It's like a comparative way. We use it to compare like one brain to another, or to compare response from one individual to another. That's what's usually you are using in the studies, right?
No, we rarely compare one brain. We usually compare group of brain to
Yeah.
increase the statistics and to be able to 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 characterize where the dysfunction occurred. But within one person, if you just put one patient in the MRI magnet, you can't yet characterize if a patient is have or not such disease. You can probably give some percentage, say, oh, this person have 60 or 17% to have this disease, but this is not going to be very accurate from as far as I know.
Okay.
Okay, thank you so much for all the questions, everyone, and for coming. And a special thanks to you, Clement, and I hope I said your name right again. And please come back anytime, maybe with one day with updates from your new job in Lyon. Maybe you will share some exciting new research. It's such an interesting field, and I think it's very important work to maybe test everything again, what we know so far from animal models and different models. And yeah, thank you so much for coming, and I hope you enjoyed it. And please, yeah, everyone in the room, follow the club if you liked discussions like this. Feel free to come back and ask questions anytime. And yeah, thank you so much again, and good luck going
Thank you for
back
your
home.
time,
I
Doctor.
hope
This was
you have
amazing.
fun.
Yeah, thank you for inviting me. It was a pleasure to speak with you guys, honestly.
Yeah, enjoy France and Europe.
Yeah, definitely. Thanks. Fascinating data. Very interesting topic. I can't wait to hear what you do next.
Thank you very
And
much.
we're going to wish you all the funding as well as all the best.
I love fun.
That's my problem at the moment. Thank you, Jamie. It's exactly what I'm doing.
Thank you. I
Thank
miss
you so much.
Thank you.
and the least bureaucracy also. And I hope those helmets work. Maybe you'll try cats. Let me know.
Yeah, it's in the back of my mind.
Thank you. Okay. Okay. Thank you, everyone. And have a good evening or morning or wherever you are around the world. And yeah, enjoy the rest of the week. And maybe you'll come back to our guest speaker events here. Thank you.
Thank you very much.
Okay, I'll close the room in 3, 2,
Thank you,
1.
Doctor.
Bye,
Good night,
everyone.
everyone. Good night.